All right, so we went over amino acid structure, the basic structure of what this looks like. Now we're going to, and that's called primary structure, is where we take amino acids and start to link them together. So if we have amino acid number 1, so it's amino acid, joined with a peptide bond, so that's the peptide bond. To amino acid number 2, 3, 4, 5, 6, 7, and so on. That's our primary structure, the specific sequence of amino acids. But amino acids don't exist in just these long, straight chains. What happens is the amino acids start to interact with each other, and then they take on shape. So at the second level of structure, right, so we call the secondary structure, we have two characteristic shapes. Those two characteristic shapes are referred to as alpha helix and then beta sheet. We're going to focus here on alpha helix right now, and then we'll talk a little bit about beta sheet. So an alpha helix, all right, a lot of times we'll have a structure, we'll see it when we draw membranes. So we'll have a membrane structure like this, and we'll see a protein is going across the membrane. And you'll see this a lot, and there'll be a protein, some complex structure, but then almost always a very regular repeated looped sequence like this, sort of like a corkscrew. Okay, so alpha helix is going to have this corkscrew shape to it, like that. What we want to see is, how does that occur? Well, what's going to happen is that the amino acids are going to start to interact with each other, okay, and form bonds. Specifically, hydrogen bonds. And these are going to be hydrogen bonds between the amino and carboxyl groups. All right, now that's going to be the key, okay? So for primary structure, we have peptide bonds between amino acids. That's the first level. Secondary level, we're going to have hydrogen bonds. Okay, and that's going to be also between amino and carboxyl groups, but it's going to be between about every fourth amino and carboxyl group. So what I've already drawn over here is this sort of structure, and right now it kind of looks like this sort of a jumble. But what this really is a representation of this alpha helix corkscrew and how it's put together as the amino acid chain is sort of twisted and interacts with each other. So what we get is something that looks like this. Okay. Here you have a single amino acid. Okay, so we're not drawing all the parts of the amino acid here, but you could see represented, I did it with color. As you can see, that's the central carbon. Here's the carbon of the carboxyl group, right? And here's the nitrogen of the amino group for that particular amino acid. And then here's the next one here, nitrogen, carbon, nitrogen, nitrogen, carbon, nitrogen. You can see these are the amino acids. And what's going to happen is that we're going to get then attractions, okay, or hydrogen bonds between carboxyl and amino groups. Okay, so there's going to be one, let's say, that goes like here, like this. This one should have, well, this should be one over here. We're going to get these alternating bonds, put one right in there, that are going to then stabilize this structure. Okay, these hydrogen bonds will make this particular coil, okay, so like this, of what we call the alpha helix. Sometimes it's written as this with the Greek, lowercase Greek symbol for alpha. Helix. Now why is that important? Well, one, it's just a generic characteristic of this particular level of structure of a protein. It would be a binding site. It'd be something that could bind to other things. And it's often the area that is found in the middle of a phospholipid bilayer in the cell membrane. We're not drawing the R groups here, but the R groups are then what would give an alpha helix a particular characteristic. So if we had amino acids embedded within a lipid bilayer, most likely their R groups are going to be those nonpolar R groups. So it'll be sticking off of those central carbons will be those nonpolar sections, which will mix in with the fatty acid tails, if you remember, which are all carbons and hydrogens and nonpolar. Okay, so protein structure has all these levels, 4 levels of structure. Primary is the sequence of amino acids joined together with the peptide bonds. Secondary is going to be a unique and specific type of folding stabilized by hydrogen bonds. The hydrogen bonds are always going to be between amino and carboxyl groups. A common characteristic type of secondary structure is called alpha helix. It looks sort of like a twisted corkscrew where the amino and carboxyl groups of about every 4th amino acid hydrogen bond with each other, and that creates and stabilizes that particular structure. All right, so that's the alpha helix. Now, as far as the beta sheet structure goes, right, so we're going to erase a little bit of this. What we're going to see is for beta sheet, without drawing all the complexity of this, so now we're going to come here. For beta sheet, what happens is that instead of the coil, all right, we have a more flattened linear structure that sort of zigzags, okay, like this, with each of the amino acids kind of going back and forth like that. This is called a beta strand. Okay, this is the beta strand, like that. And there's not really specific hydrogen bonding in here. It's just amino acid with a peptide bond to another amino acid. Instead of being straight like this, or instead of being curved, it's a zigzag. That's what we're seeing. What happens is that at a certain point, there's a loop. It's called a random coil, which is because of the amino acid proline. And it allows multiple beta strands, like this, to kind of fold back on each other, and so on. Now what happens is that the amino acids in these strands then form hydrogen bonds with the amino acids directly across from them. Right? It's still going to be hydrogen bonds between amino and carboxyl groups. So, regardless of whether we're talking about alpha helix or beta sheet, when we're talking about secondary structure, what creates or stabilizes secondary structure is defined as hydrogen bonds between amino and carboxyl groups. All right? How and where they form gives us one type, the coiled alpha helix, or the zigzagged beta strand, which when the strands join together, this is now called the beta sheet. So a sheet is many of these together. It is sort of similar to the way a piece of paper, if it were folded, the reason it's called sheet, if you had a piece of paper and it were folded like this many times, back and forth, this sort of zigzag. Okay, you can kind of line it up. The zigzags, right, of the sheet, we're kind of representing this. But if we look at it sideways, it's actually this flat, all right, plane of the piece of paper because of the way they would line up. So they create these sorts of structures within the protein. The significance of the beta sheet is that there are many hydrogen bonds. Which makes this structure very stable. It's incredibly strong. So the protein, for example, found in spider silk, so in spider webs, it's made up of protein. That particular protein is full of beta sheet structure. As a result, it has 10 times the tensile strength of steel. What does that mean? It means that if you have a string like this of spider silk and another string like this of steel, and they were scaled to be exactly as thick as one another. So you either made the spider thread thicker to match up a thread of steel, or you made the steel really, really fine as the spider silk, the spider web, and then you hung weights from them like this. You could hang 10 times more weight from the spider silk than you could from the steel. Right? The steel would break, right, before the spider silk would. And it is that strong because of the hydrogen bonds. Now, then sometimes people get confused because they say, aren't hydrogen bonds weak? Yes, an individual hydrogen bond is a weak bond. But here you have many bonds. The bonds don't themselves get stronger, but it's the collection of them. It's that there are many bonds. There are so many of them that collectively, if you add them all up, it makes an incredibly strong and incredibly stable structure. And this is something that people research and are trying to develop fabrics and materials, specifically with proteins that have a high amount of beta sheet structure in it, because they are so strong. And this is kind of an example of where that comes from. So at this point, what we need to know or be familiar with the terminology. Two terms, alpha helix and beta sheet. They are two subtypes of secondary structure of proteins. They are both stabilized and are created by hydrogen bonds forming between amino and carboxyl groups. Right? So that's partial positive attracted to partial negative. So typically, the partial positive here, hydrogen, is going to be attracted to the partially negative oxygen. That's what's going to happen. So it's always going to be this hydrogen-oxygen, hydrogen bond, and there's going to be attraction between the two. The alpha helix is the coil. We find it in a number of places. It's very common within many proteins, and we see it across membranes. The beta sheet structure is a zigzag, and that ends up folding together in a particular way that makes it very, very strong because of the large number of bonds. Right? Tertiary structure is going to be far more complicated, than this. So all we're going to give you is a few specific rules and definitions. I'm not really going to be going much into drawing of the structure for those. And the same with quaternary. That is a much higher level of structure. It's where we have multiple polypeptides, hundreds, thousands of amino acids joined together, already folded and twisted in a variety of ways, and then they have to join together. And that joining is going to be the quaternary structure. So we're not going to be drawing those high levels of structure. Again, for you should know amino acid structure, the definition of really basic primary structure, peptide bonds. You should be able to do a little squiggle, something just like this, and just say, hey, that's my alpha helix. You would not have to worry about this. So do not worry about trying to draw all the amino acids and how they join together like this. It's just sort of an example to put the chemistry and the elements behind the little curly structure to kind of see how they would come together. And the same for beta sheet, just be able to recognize it. You should be able to just sketch out something with a little zigzag and join them together. Again, hydrogen bonds are what holds them together. If you know that, then you know all you really need to know for this part of the material.